Hello, my name is Jacqueline Medina, and I'll be presenting over experimental and computational NMR strategies to identify constrained conformers of modified cholecystyrene. Cholecystyrene is an organic molecule composed of 4 parahydrocarbonyls linked by methyl groups. The cholecystyrene form we'll be looking at contains a tert-butyl group in the pair position to the hydroxyl group. This molecule has multiple applications, such as enzyme mimetics, in which cholecystyrene is used as a catalyst to replace enzymes, ion-sensitive electrodes, which enhance accuracy by minimizing matrix effects, drug delivery, where current research is studying the transportation of anti-cancer drugs, and as well as ion binding. A deeming quality of cholecystyrene that facilitates that transportation is their rim-like structure, in which a cavity is contained inside which serves as a carrier. Due to this, cholecystyrene can be described as a chaperone molecule. Multiple isomers of cholecystyrene can be produced, which are shown as cone, partial cone, 1,2 alternate, and 1,3 alternate. Differentiating between these 4 conformers can be a challenge, but with the use of computational NMR, conformers can be identified, in which the conformer focused on this study will be cone. For schematics here, bis-ethanolamine will perform a nucleophilic aromatic substitution on para-nitrofluorobenzene, which replaces the fluorine group. The hydroxyl groups are then activated by tosylating them, which once purified will be reacted with cholecystyrene, which results in multiple conformers, 12% being the desired cone with the linker group. This yield is hypothesized to be low due to bis-ethanolamine's anti-periplanar nature, further leading on to elimination, in which the lone pair on the tosylated form will react with itself due to it containing a good leaving group. Due to this, not only is it decreasing the yield, but as well as creating side products, which further react to the multiple conformers, which further challenges the separation due to similar polarities. With the use of NMR, similarities are seen between both the conformers 1,3 in the top spectra and cone on the bottom spectra. Due to this, different characterization methods will be used in this study. For 2D analysis, nuclear Overhauser effect spectroscopy, known as NOESY, distances between interprotons may be determined. These interactions are displayed in the XY cross-peak spectra. Based off the spectra above, possible conformers could be 1,3 or cone due to data being symmetrical, as is both molecules. It could be possible that this conformer is cone due to the presence of the phenyl-nitro interactions with the hydroxyl group. So basically, this proton interacting with the hydroxyl proton is seen on the cross-peak of 6.91 and 6.45, and we also notice that there's an absence of phenyl-nitro interactions with tert-butyl. Although promising, this data is inconclusive to determine conformations because no other conformers were used to compare. Due to this, rotating frame Overhauser effect spectroscopy, known as ROSY, was performed in order to calculate proton distance through space. Using computational modeling, DFT, methylene distances between phenyl protons, so bridged protons were compared, interactions with phenyl protons were quantified, in which ROSY matched both reference DFT and X-ray literature data with a length of 2.39 angstroms. In this figure, the cone conformer can be characterized based off methylene phenyl proton distance interactions, which differ greatly compared to partial cone and 1,3. As you can see here, the 1,3 alternate and the partial overlap, while the cone depict a shorter methylene phenyl distance. The gray bar depicts the analysis range using ROSY, while the data exceeding was determined computationally. Something to note is that these protons are not symmetrical, which is why 4 different proton signals are visible in red. In other words, cholecystyrene with the linker group has more of an oval shape rather than the expected circular shape with 2 proton signals. For computational analysis, predictive NMR is developed using conformer and ensemble sampling tool, known as CREST, in which carbon and proton NMR corrected mean average error, CMAE, data are combined. Computational chemical shift values were compared to experimental values, which were then graphed and compared, as you can see here. When combined, cone has the lowest CMAE. Looking at the graphs, it's not as prevalent looking at just solely the carbon NMR. By combining both of these, you're able to see that cone has the best fit. In conclusion, ROSY served as a great quantifying and qualitative method for proton-proton distance, and computational analysis of carbon and proton NMR further support the identification of cone. I would like to acknowledge and thank our collaborators and as well as the NSF Welch Foundation for funding this study. Thank you.